So I had the fortune to meet Lorne in 2016, so 4 years ago. He came through the door, was interested in global health, and was eager to start participating in the work that we do, and also in looking at how ecology and health intersect. So Lorne jumped right in, and we were able to plug him into a bunch of projects that shaped all of us over the subsequent 4 years. It's hard to believe that 4 years have gone by already, but looks like it has. So so everybody knows, Lorne comes into the University of Rochester with a master's degree in environmental science from our neighbors at RIT. And before that, with a dual bachelor's degree in biology and chemistry from Gallaudet University. And Lorne's PhD with us is in the Translational Biomedical Science, or TBS program. And we have a special track devoted to infectious disease and community health called IMP. And Lorne was one of our students in the IMP program, which meant he had a special emphasis on infectious diseases and population health. And in Lorne's case, that extended into global health. So Lorne, for those of you that know him, just jumps right in. And so we started off with some work in Costa Rica, and Lorne participated as one of the faculty members in a training program that we had for NIH. And quickly, it was clear the value that Lorne brought to the team. First of all, he has experiences that others don't have. He has a certain insight and interest in ticks, which we didn't know anything about. And in most of the countries where we work, they didn't know anything about. Yet all were dealing with the consequences of ticks. So in Costa Rica, Lorne ended up being one of our faculty members on our training program. And along the way, we were able to engage with a number of deaf communities and deaf populations that we still work with. Lorne traveled with us several times there as part of our team. We have a rumor that this dog is missing, so I hope, Lorne, you didn't stick that dog in your suitcase on the way back. The animals kind of gravitate to Lorne, so that's one of his special talents. Another place where we worked is in the Pacific Islands, and this is a very unusual part of the world for people from this part of the world to work in. And we are very fortunate to work in a very remote set of islands in the Northern Marianas and in Palau. And Lorne was also pursuing his tick interest, but he was working on our project looking at chronic disease. And these were very remote areas. We flew in these tiny airplanes, which was nothing for Lorne because he skydives and jumps off mountain sides or whatever else he does. But this airplane, I think Jackie was holding the door as we were flying in the air. You know, for the rest of us, it was a little bit nerve-wracking, but wonderful nonetheless. Lorne very quickly engaged with the community there, took an interest in the history, and very quickly was just a core part of the team. So that really was a testament to his ability to bring his research to all kinds of people and to just quickly integrate with the team. Though Lorne is deaf, and this is certainly a positive characteristic in our work, where we can now reach deaf communities that we were never able to reach. Most of Lorne's work was with hearing communities. So he interviewed people in the Pacific Islands very well. He was a very good interviewer, and people participated very eagerly in the work that we did. And the fact that Lorne is fluent in ASL really helped us be able to explore issues and reach communities that we otherwise wouldn't have been able to do. Lorne picked up some skills along the way, I'd like to think. Coconut tossing is one of those skills that he's probably better at than the rest of us. So glad to see that we ended up teaching him something other than epidemiology and field research. And this picture takes some explanation, and I'm probably not going to explain. I'll let Lorne decide if he wants to explain what this picture is or not. Yes, that is a bat, and this is Palau, and I'll just leave it at that. And Lorne can tell you that story if he feels like telling you that story. In Costa Rica, you know, one of the things that was really interesting about Lorne's participation was that in Costa Rica, the deaf community uses LESCO. So we pretty quickly found ways of integrating LESCO. We had Costa Rican deaf participants. We had Costa Rican Spanish-speaking hearing participants, and we had English participants in this project there. And Lorne and several other people being ASL fluent. So we had all these languages going on at the same time, and it really gave us a wonderful experience in communication and how important communication is and how important it is that we figure out what's needed so everybody can fully participate in what's going on. So this was just a great example of all of us being able to participate with all the communities that we were working with in a very, very fluent way. And of course, we're very grateful for all the interpreters that have worked with us along this journey as well. So all this has led to a considerable amount of productivity. Lorne has a number of publications. He's first authored publication on one that came from his dissertation research. Lorne's eagerly participated in the academic life of our TBS program. He regularly gives guest lectures and courses. He met Dr. David Satcher when David Satcher came, and usually participates with all of our recruitment activities. So Lorne has been a great citizen of our TBS program and of the university, and very productive along the way from all these different aspects, most recently COVID, of where he's brought his expertise to bear in our science. Lorne's also done quite a few conferences. Again, since we're a global group and Lorne's a global person, most of these conferences have happened around the world. And he's worked with a number of our other TBS students and IMP students along the way. So he's a very important part of this, this family that we have. So without further ado, I would like to turn it over to Lorne, who will present his dissertation, Surveillance of Global Tick-borne Disease in Ecological Context, Geographic Distribution, Novel Bio-diagnostic Testing, and Preventive Strategies for the Deaf. And I'd like to ask that people refrain from asking questions during the presentation, and after Lorne is finished, we'll be able to entertain questions at that point. So Lorne, I'm gonna stop sharing and it is yours.
All right. Hello, everyone. Thank you for taking the time to join me today as I talk about my exciting findings in the research that I've been doing in tick-borne diseases. Thank you so much for Tim for that wonderful, beautiful introduction. Very heartwarming. Before I begin, I wanted to recognize the many wonderful people that have helped me along in this research. Without them, this would be impossible. I'm humbled and honored to work with two fantastic co-advisors, Dr. Tim Dye and Dr. Ben Miller. They've worked together to support me in the two disciplines I've been studying, foundational or basic research, and public health and population health research. And thank you so much to my committee members, Dr. Brian Ledette, Dr. Carol Patton, and Dr. Edwin Van Meijden. They've also provided a lot of support, advice, and guidance on which directions I should take. There have been 4 labs that I've rotated amongst over the course of my 3 projects, but all that hard work has paid off. So thank you so much to friends and family, also my interpreters that have worked with me and suffered through me and managed huge terms and technological terms with me as they interpret from English A as well. Also, a brief note before I start, I do have 2 interpreters here with me, Jacqueline and Valine, and they'll be interpreting for me from ASL to English. You might recognize some pauses during the course of the presentation, and that's because of that interpreting time and also to allow deaf viewers to both see the slides and then turn back toward me. So I'll be discussing tick-borne diseases and why they're a problem. There are many reasons and many variables involved in why tick-borne diseases are growing in prevalence. Tick-borne diseases happen everywhere in the world. Ticks have the capacity to spread over 15 diseases, or 13 diseases to humans. One common disease that you may have seen here in America is Lyme disease. Other ones here are Rocky Mountain spotted fever, anaplasmosis, and a new pathogen recently discovered a few years ago called Powassan virus. That was identified in Canada, and the concern is that it's been spreading. Where diseases emerge depend on a variety of different factors. The maps here show the ranges for anaplasmosis, babesiosis, usually in the Northeast. And these incidences of disease really depend on the tick species involved. And each tick species usually has their own preferred host that they'll draw from. Climate is another important factor here. There are many variables involved in tick-borne diseases and how they continue to spread. I've included some of them here. Environmental disturbance and land use changes, such as agriculture and deforestation, are changing the landscape and have a strong effect on ticks and where they migrate in and out of, as well as where their hosts migrate. Climate change is a major factor here. It's also important to consider where ticks are emerging in relation to their hosts. Ticks there are over 900 species of ticks. Each tick species has their own preferred host and range. For example, Rhipicephalus guophalus microplus is a tick that almost exclusively parasitates cows and does not bite humans. Another example, Ixodes, is often known as the deer tick, but does bite humans. So there are many different ticks and many different niches for them, but we are seeing the incidence of these diseases increase. Here are a number of other variables involved. Pathogens play a large role in this. Why a disease might appear in one area and not another. There are a variety of factors there. Mode of transmission needs to be considered. There can either be anterior transmission, meaning through the mouth, or posterior transmission, meaning bacteria spread through the feces. There's also horizontal and vertical transmission being from the parents, whereas horizontal transmission would be through different life stages, larva, nymph, and adult. So again, a number of variables, and a very important one here includes societal, economic, racial factors, including access to the healthcare system. What role do they play in tick-borne diseases and their spread? So with that large number of variables that we just saw, it seems overwhelming. But with this framework, the medical ecological model, or MEM for short, it's a wonderful platform to be able to put these together and see where the problems are and what needs to be studied. So in this case, I used tick-borne disease as the outcome variable. And this really helped me take a step back to see what roles these different factors have in 4 different domains: sociocultural, biological, healthcare, and abiotic, all on 3 different intervals: individual, household level, and community. And that helps us understand where to prioritize our efforts, what factors are involved, where research needs to be developed, what equipment needs to be utilized, what skills need to be brought to bear. And all of that helped me with developing this project. Using the transitional research approach, this really helped me to see where the ticks are in regards to tick-borne disease. The translational research approach will help me put everything together for this project and see how to leverage that to improve healthcare infrastructure with regards to tick-borne disease. The translational approach uses 2 different fields or disciplines and merges them together to support each other. And that involves foundational or basic research and public health and population research. So this transdisciplinary approach merges two different fields and allows me to get trained in both of them. So this project is comprised of 3 different projects: the epidemiology of ticks and tick-borne diseases, preventative strategies for the deaf population, and a new diagnostic test. These 3 projects are things that I've been working on concurrently for the past 3 years. This is my first team. The study of ticks and tick-borne and their distribution geographically. My first name is here. And the hypothesis is that the prevalence of infection in ticks and the risk thereof varies by region. So this will help public health and clinicians to develop the proper approach to combat tick-borne diseases. So again, the hypothesis here is the prevalence of infection in ticks varies by region. So one of the many factors influencing the rise of tick-borne diseases is, again, land use changes and agriculture and deforestation. Also, the migration of birds and animals. As they move to different places, they contribute to the rise of tick-borne diseases as ticks move with them. Also, there's a lot of evidence showing that climate change plays a huge role in the spread of tick-borne diseases. Evidence shows that over the course of the past 100 years, there's been a 2 degrees centigrade increase in temperature worldwide. It may not seem like a lot, but it has a huge influence on things going on around the world. That temperature increase causes ticks and their hosts to move to historically or to higher elevations or northward geographically in the northern hemisphere. Areas that were previously colder are warming up, allowing ticks to settle and spread into new areas. This example here shows a map of the US from 2001 to 2015 and incidences of Lyme disease. So each dot represents a case. If you compare 2015, you can see the huge increase in number of cases and where they're appearing. The expansion of ticks is going to lead to a greater incidence of disease. In terms of tick biological processes, they spread pathogens through their mouth parts. They'll be aggressive towards the host that they prefer, but their behavior can also change as a result of climate change. And we'll see an increase in the incidence of tick-borne diseases. Ticks transmit infection through the capitulum, for their mouth parts. As those pierce the animal and they start drawing a blood meal, they will also regurgitate back into the host. And various pathogens that live in the gut of the tick will be regurgitated in the host's body and cause a systemic infection. Another issue is that diagnostic testing for tick-borne diseases is often unclear and problematic. Tick-borne diseases manifest as a number of different symptoms that can be unclear to physicians. And also, a lot of these diseases are serologically similar, so that can cause cross-reaction with diagnostic tests, making diagnosis and treatment more complicated. A previous study that was done analyzed 222 serum samples from people in Ecuador. They were testing to see if those samples were positive for rickettsial infection. They detected 18% of those 222 serum samples were positive for rickettsial infection. That's a concern to see that number of disease in the country. Ecological epidemiology is an important first step to understand these diseases and what role they play and how they affect us. To date, there's no documented cases of pathogenic rickettsia in Ecuador, of bacteria known as rickettsia in Ecuador. Spotted fever group rickettsia is not part of Ecuador's National Disease Surveillance Program, which means it's a concern that there might be an actual burden of disease there that's being neglected. That country is facing serious health consequences. So that's why I felt it was important to go to Ecuador and collect ticks for this project. Ecuador is known as one of the most ecologically diverse places in the world. It supports 12 different life zones. The country is woefully understudied with regards to ticks and tick-borne diseases, so I saw the need there. These videos on the slide show the variety of ecologies in one country. It's a beautiful place. This was driving from the coast, through grassland, through rainforest. You can see the number of different biomes that one can pass. Again, it's a beautiful country. Ecuador is in South America, next to Peru and Colombia. Ecuador is comprised of 24 provinces, which are similar to the United States here in the States. El Oro is one of those 24 provinces. El Oro is divided into 14 cantons. El Oro is known for its historic gold mining, its history of gold mining. Later on, it became one of the top banana exporting countries. This has caused a lot of environmental disturbance and changes in land use, which supports ticks moving and expanding their range. So I chose El Oro as my study area. And I chose 5 different cantons, which were then grouped into 3 different ecozones. They were grouped based on their similarities in temperature, humidity, and altitude. The goal of the study was to understand if there's a distinction between tick infection and environment, comparing those 3 ecozones. Now I wanted to talk about the methods employed in the study. I collected ticks 2 different ways. I collected free living ticks from the environment. Free living ticks mean that they're in the grasses, they're climbing to top of blades of grass, and then they use a behavior called questing, where they're searching for a host. I used a method called flagging and dragging, which uses a white piece of flannel and is dragged over the grasses, and the ticks will latch on to that cloth as it drags over them. The second method was that I collected ticks off of parasitized farm animals, such as cows, horses, and dogs. Once the ticks were collected, I identified their species. This was based on their morphological features. The main morphological features for ticks to help distinguish species are the scutum, which is the tick's back or shield, the festoons, which are the bottom spots along the back, and the capitulum, which are the mouth parts. The mouth parts can have different sizes, widths, lengths, and that helps to distinguish tick species. After they were identified, I extracted their DNA and then ran PCR experiments, polymerase chain reaction experiments on them, which is a way to amplify DNA and see if those ticks are test positive for any of the pathogens that we're looking for. For statistical analysis, I used a bivariate analysis, and my variables were all categorical. And I used an alpha of 0.05 to see if there was statistical significance there. The second method I employed was, I figured an odds ratio, which is the odds of finding an infected tick versus finding an uninfected tick per ecozone. Then I ran a multivariate logistic regression. And for this, I used Hosmer and Lamashow's purposeful method, which helps to detect if there exists any confounding variables in the model. The slide shows the dependent and independent variables that were used in the model. And then we're looking to see the odds of finding a tick infected with rickettsia. So of the data I collected 372 ticks in total. And 5 different tick species were identified. The species are Amblyoma cahinense, Amblyoma ovale, Amblyoma maculatum, Rhipicephalus sanguineus, and Rhipicephalus huophilus microplus. 5 species were identified. And then these were collected across the 3 ecozones on the top, as you can see. And where the p-value equals 0.05 or less, it's considered significant, and you can see that highlighted. So you can see I found a large number of Amblyoma cahinense, and more so in ecozone 2 versus 3. That was the most significant and interesting finding, but there is more. So tick sex can be broken down into male, female, and non. I also broke this down into tick host type, again, horses, dogs, and cows. I collected mostly adult ticks. And most of the ticks that I did collect were from cattle. So now to test if those ticks were infected with rickettsial bacteria. 42 of the ticks collected were found to be infected with rickettsia. On further analysis, we used 3 different primers for that, the 16 kilodalton primer series. The second and third primers were focused specifically on the particular bacteria that we were looking for. So we were able to identify 2 kinds of bacteria in the rickettsia family, R. parkeri and R. emblematis. One of the largest samples that we received, actually the largest sample that we collected, was Rhipicephalus huophilus microplus, but it is solely a cattle tick and does not bite humans. So that was of lesser concern and was excluded from the study in the analysis. So from then on, the analysis focused on Amblyoma cahinense, which is the second largest tick and the tick with the largest number of infected ticks. So this is what we'll be focusing from now on. Now to see if environment or ecozone has a role in tick infection. So in comparing the ecozones, with 95% confidence interval between 1.8 and 8.7, there's a 4 times greater chance of finding a tick infected with rickettsia in ecozone 3 versus ecozone 2. Now for the logistic regression model. We wanted to identify any possible confounding variables in the study. Model 1 here was only 1-to-1 variable. So 1 variable versus another. And you can see the odds ratio there of 4.0. Models 2, 3, and 4 added or subtracted various variables to see if there was any major change within 10% of that initial 4.0 odds ratio. If there was a change greater than 10%, it showed that that variable was a confounder. So you can see the list of 4 models here and their various odds ratios that were all within 10% of each other, showing that there's no confounding happening in the study with these variables. In conclusion, we found 5 different tick species Of those ticks, many were infected with one of two rickettsial species. And the data study shows that the incidence of infection does in fact vary between ecozones. And the most important finding here is that finding of Rickettsia parkeri that was found in 3 ticks down there. And that is of concern. It would be important to do a literature on rickettsiosis in Latin America. After conducting a literature review myself, I realized that there is rickettsia in Ecuador, but it's Rickettsia prausechii, which is only transmitted via lice, not ticks. Therefore, this is the first documented case of a tick infected with Rickettsia parkeri that could potentially cause rickettsial disease in humans. The limits to this study are that we use 2 different collection methods for collecting ticks: flagging and dragging, and collecting them from parasitized animals. To have a better study, I would have just used one method, because having 2 introduces sampling bias. Also, questing ticks, those free-living ticks looking for a host, are often less likely to be infected with different pathogens compared to ticks that are parasitizing animals. They often get infected from the host they're feeding on. So those ticks that we collected, those free living ticks, are not an accurate reflection of the total number of infectious ticks in Ecuador. Another limitation, this is a one-time study, it's one data point. So one point of collection is not long enough, there's not enough data to see long-term changes, such as from climate change or a tick's life cycle, which can be about 3 years. So we need at least 3 years of study to really have a true reflection of what's going on with ticks and tick-borne diseases in the country. This was a small cross-sectional study, so findings cannot be generalized. Scientific contributions from the study is understanding and describing ticks and their associated pathogens among several locations in southern Ecuador. Future directions for this work would be to share our findings with the Ministry of Health in Ecuador and make recommendations to possibly include rickettsial disease in their national surveillance program in consideration of the pathogens that we found. And so that if patients are infected with something, that would be a consideration. They could have a tick-borne disease. Also, further research on the pathogenicity and virulence of the pathogen called Rickettsia embryomatosis. There's some controversy there about whether it's actually pathogenic to humans or if it's in fact an endosymbiont, which seems to be the prevailing view. An endosymbiont would not cause disease to humans, but there does seem to be some cases of people who are bitten by a tick in Ecuador and do seem to be infected with that bacteria based on their antibody counts going up. So we're not sure if that bacteria in fact has a role in disease. That would take more lab work, more lab research into that. So now, my second aim. So this aim is looking at a new diagnostic test to see that it can be applied to the detection of tick-borne diseases. My hypothesis here is that 2 diagnostic platforms called arrayed imaging reflectometry, or AIR, and grating coupled fluorescence plasmonic, or GCFP platforms can detect antibodies to tick-borne diseases in 3 diseases: rickettsial disease, anaplasmosis, and Lyme disease in blood serum samples. Tick-borne diseases and testing for them, well, there are many tick-borne diseases, but often their symptoms look very similar. So diagnosing these is tough. Ticks can spread over 13 different diseases. Also, ticks have the ability to transmit 2 or even more diseases, also known as co-infection, which can make a person's disease burden worse. Right now, diagnostic testing involves a number of different steps, such as ELISA, IFA, PCR, blood smear, and cell culture. And those lead to a complicated algorithm, as you can see here. There's many steps involved, it's time consuming, and it is expensive. If someone's being tested for a specific disease and then they test negative, then subsequent tests with other diseases need to be given, which increases the time it takes for diagnosis. So my proposal here is that these technologies can combine several different tests for tick-borne diseases on one platform. So along with a record of symptoms, AIR or GCFP can detect a tick-borne disease directly to make sure it's identified properly and what kind of bacteria is causing it. After that, the patient can be given the proper treatment plan. AIR and GCFP are very similar technologies in how they detect disease. They each involve 3 main steps: printing, processing, and imaging. There are 2 different types of chips that can be used that are printed on: silicon dioxide, or SiO2, or gold for the GCFP. They're almost similar in properties and chemical composition, and they help to detect disease. First antigen is printed on a chip using an instrument called an arrayer. It's a high-tech instrument to deposit a very small amount, about 200 picoliters in a 100 micrometer dot on the chip. So it's a very small amount, but it prints many, many, many dots on the chip. So this way, the hope is that it would run many samples for different diseases at the same time. Once printing is done, it's processed with a specific analyte, depending on what you want to analyze, either an antibody or antiserum. Antibodies are what our system, what our bodies, what our immune systems make to defend ourselves against diseases. And these antibodies are very specific to a particular antigen from a bacteria in this case. AIR only requires a primary antibody to be used. Once the antigen is printed, the antibody binds to it. GCFP requires the use of the primary antibody and the secondary antibody, which is fluorescently labeled and will emit energy if it's excited by the binding reaction. GCFP, here you can see how it requires that second antibody to fluoresce and indicate if binding is occurred. AIR requires 3 fixed variables, which is why it doesn't need the secondary antibody. It needs to be a proper wavelength of 632.8 nanometers, particular polarization of light, in this case S polarization, and the angle of incidence, which must be 70.7 degrees. AIR also depends on the use of a helium neon laser. So I tested 3 diseases. Each have their own antigens and antibody pairs. And I chose these diseases based on a need-based approach to see where the greatest prevalence of cases are occurring and where the priorities should be. So this is testing Lyme disease, Rocky Mountain spotted fever, and anaplasmosis. So the methods in this study incorporated 4 different experiments: a dot plot experiment, SPR, AIR, and GCFP. Starting first with the dot plot. It might be the most basic approach, it's just to detect if binding occurs or not. It uses PVDF membrane, where a small amount of antigen is put on the membrane and then dipped in an antibody to see if they bind. I use anaplasmosis as an example here, and you can see the reactions on the slide. At the 100 microgram per milliliter concentration, you can see a faint reaction happening up on the top corner with TSG. With the FDWN peptide, its antigen did not bind. Second, using SPR. The goal here was to see if we were to add streptavidin to the experiment, if results would improve. SPR is a biotinylated peptide, which has a strong affinity for streptavidin, and it really facilitates the interaction of molecules to help binding happen. So the binding, or the interaction, was between a biotinylated peptide, streptavidin, and the antibody. Biotin covalently binds to a protein. So biotin has a molecule that has a high affinity for streptavidin. Streptavidin helps with the interaction between the molecules and helps stabilize the reaction. So adding streptavidin along with the antigen antibodies should hopefully encourage that interaction and stabilize the reaction. But first we need to see if streptavidin, the antigen, and the antibody can be used. And these diagrams just show how that occurs. First, the chip is coated with streptavidin, the antigen is added, then the antibody. The results here show 3 different cycles. The first cycle is the control. There's no antibody or antiserum in that part of the study. So that simply serves as a control. The second and third cycles are where the antiserum was added. And you can see how the peak increases from there to show the binding has happened. Cycles 2 and 3 have a reaction response unit along the side there to see and quantify just how much binding has occurred. So you see that 950 and 762 for cycles 2 and 3. So with the first 2 experiments confirming that binding can happen, we wanted to move on and test air to see if that could be used as a platform for detecting tick-borne diseases. I started first by figuring out what chip thickness was ideal. There are 3 different thickness levels, as you can see here, and it's important to find the proper thickness level to establish near zero reflectance. Then this way we can know if binding is happening or not. Results showed that the ideal chip thickness used for this study was 1378 angstroms, so the middle diagram here. Next, an analysis was done to see if anaplasma and lime could bind in air. There was a range of reflectance change shown here. You can see the rectangles outlined there to show where binding is happening. Anaplasmosis, or its associated antigen, showed a little bit of reactivity, a small change in reflectance, about 0.5. Lyme disease, or the pathogen Borrelia burgdorferi, its antigen, showed a very small change in reflectance, just in one spot. But overall, it did show some weak reactivity, but the results weren't overall positive. Now on to GCFP. I used this to test for anaplasmosis first. And the green squares there are examples of the control. The array layout here, next to it, corresponds to those green squares. But in those white columns, it means that no reaction is happening. And same results for anaplasmosis. We didn't see, or its associated antigen, we didn't see any response. Finally, we did get a positive response using Lyme disease. So in conclusion for this study, air did show a weak reaction and a reflectance change. And also 1% trehalose can be added to the chemicals that are printed with the antigen. So hopefully spread out and provide more uniformity of the printed dots. So 1% trehalose is found to be an ideal additive. And then for GCFP, we did see a strong reaction happening with Borrelia burgdorferi pathogen associated with Lyme disease. So this research needs to be continued to understand the interaction between molecules. Maybe if a larger spot was printed on the chip, if there would be more interaction and a stronger response. Also, just to add, I didn't have sufficient time to really look into this in this study and to really analyze things as they should be and should be continued. So that is one limitation for the study. So this does suggest that air and GCFP have the capacity to detect tick-borne diseases, but a lot more work would need to be done. In terms of scientific contributions, we recognize antibody and antigen interactions for tick-borne diseases. Future directions for the study would be further analysis on antigen and antibody interaction, maybe adding that second antibody that would fluoresce and give a better idea if binding is happening or not. And maybe adjusting the size of those printed dots on the chips. Now on to my third aim. My third aim focuses on preventative strategies for the deaf community. How I went about this is using a qualitative research approach, which is doing qualitative interviews with the deaf population to find out the most effective scientific communication approach with them. The hypothesis, as you see here, is the prevailing tick-borne disease preventative information is not accessible. Therefore, a tailored communication platform for deaf populations can effectively deliver tick disease prevention information. I did want to start by letting everyone know that the original plan for this aim was to do qualitative interviews with the deaf population in Ecuador. But because of coronavirus, we had to make some adjustments to the research plan. Therefore, we did interviews with people in Western New York. The data can be applicable to deaf communities in Ecuador because all deaf people who are using sign language as their primary communication experience the same kind of oppression and inaccessibility to information. We do need to be considerate of their culture, context, language, and system, which is different than the community here in Western New York. The deaf population is considered a high-risk group globally. There are 360 million people with hearing loss. That's about 5%. 70 million of those people use sign language as their primary mode of communication. There are different sign languages based on region all across the world. Here in America, we use American Sign Language. The deaf population is a unique group of people that require a specific approach in order to address their health disparities. They experience language deprivation and don't have access to language acquisition. And a lot of that information is provided through hearing, therefore they don't have that. The society historically has been focused with an audio-centric approach, therefore these deaf people don't have access to this information. If we really would like to have equal access and have the health disparities be broken down for the deaf community, we need to really start with that view. Having information not be accessible to these deaf people is a form of oppression. There was one study that focused on health literacy skills of deaf people and their peers. They found that deaf individuals were almost 7 times more likely than their hearing peers to have inadequate health literacy. A study decided to translate health information into ASL. They found that many deaf people were able to understand this information.
The clinical information system does not provide information in the primary language of American Sign Language. Therefore, they don't have access and don't understand the health information that's provided. We see that there is poor health literacy in the deaf community because the information is not available to them and is not accessible. So we just see this as a huge barrier for the deaf community being able to get access to this information. To study the deaf population and understand their way of life and how they think, we need to be able to take a step back and think of the theories that are used in this study. One of them is the health belief model. This helps us understand what the beliefs are of a person. And first we need to understand how they get that information, how they perceive problems. Do they understand the consequences? Do they have action planning that would reduce the consequences? And understanding that a benefit is better than a risk, and therefore changing their behaviors because of that understanding. The second theory I included was, after finding out what the health belief model is understanding what is the theory of reasoned action. So whatever their belief is, what is the norms or the attitudes that surround them, because that also affects their ability to adopt a health behavior or not. The third theory, which is very important, is the social cognitive theory. This is very influential in a person's decisions because it thinks about their environment and the people that they interact with, and that really has an impact on what information they get and what decisions they make. With all of these theories, we can develop the right kind of questions to then ask the deaf population their thoughts and ideas on this. The health literacy model is also important to include in this study. There are two different approaches. There's the skill-based approach, which is text English-based, and then there's the task-based approach, which is focusing on function in an interactive approach. Information is typically provided through things that we can hear incidentally, people talking in the hallways or families talking on the radio, on the TV. Therefore, deaf people don't have access to all of this information, therefore they have lower health literacy. This study specifically showed that if ASL is provided if they get information in ASL, then they understand that information. Therefore, in turn, they also will understand English better. So it actually has a secondary gain. If information becomes more accessible for the deaf population, it will help them become more health literate with different English terms as well, not just in American Sign Language. Throughout these interviews, it would help us understand what kind of materials would be best fit to have them be able to have access to this information. With the methodology, we decided to use a snowball sampling approach. Participants were 18 or older. They lived in Rochester or Buffalo, New York, and they had to be deaf. It was a semi-structured interview that was comprised of focus groups and individual interviews. The qualitative data analysis focused on a program called Deduce, which you can see here on the slide. The code book included the code, which was from the EDIC and EMIC perspective, which is the person's internal perspective, and then an external perspective. There was a brief description, a full definition, and examples. All of that comprised together became the code book, which helped us identify what are the patterns and themes throughout the interviews. Those became the data that helped us create our results. Everything was done in American Sign Language, and we needed to translate that into English, so we used the Brislin's method to make sure there was nothing lost in translation. What was important about this approach is that almost all qualitative interviews in research have a transcript. They're based on transcripts and spoken language. There are several that include sign language, which requires a whole new approach to this. With coding, we are directly coding from sign language. We're not using the transcript, the translated English, because then we lose the nuances and the true meaning from the original language. These are the results of the study. First, I have the demographics. There were 40 people that I interviewed. The average age was 33. There was 24 women and 16 men, which was almost even. There was Caucasian, Asian, and other races as well. We had a variety of education levels, as you can see here. There's also a section that talked about how people prefer to get their information in American Sign Language, or if they would rather have other approaches as well as written English. The average length of an interview was an hour long. There was 16 individual interviews and 5 focus group interviews, and no one dropped out. The themes that I acknowledged with the results are listed here. The one that was the most prevalent was the perceptions within the deaf community and deaf culture. I can give you a couple of seconds to read the slide, because this is an important quote. Within the deaf population, there is a divide. We have those who are considered grassroots, which people would say they are real deaf. They are not colonized by the audiocentric hearing society. And then there's the highly educated deaf community. In Rochester, we notice there is this huge divide. When it comes to health information or content that's available, we see it's geared more for the highly educated deaf population than the grassroots. They do feel excluded and not included. There's terminology and words that are used that they have not been exposed to and don't understand. Another issue in the community is the digital divide, which suggests that deaf people who have a low SES and cannot afford technology don't have access to this information. And it's really withheld from them, because how else are they going to get it? In today's age, we see it, technology is the main way of people communicating this information. So there are complex layers of obstacles faced by deaf individuals. In an audiocentric society that relies mostly on being able to hear, deaf people struggle. A lot of society wants deaf people to be fixed, which is based on a stigma that deaf people are broken and need to be helped. They're viewed as a way to earn money in the medical system, and there is no collaboration with public health sectors and the deaf community and understanding the community and culture and language that they have. And it is an invisible disability, so they are typically neglected. In regards to ticks and tick-borne disease awareness, we saw there were some deaf people that knew a little bit and some that knew nothing at all. It's concerning because there is no information available in American Sign Language about ticks and tick-borne diseases. We need to be considerate of deaf people's culture, their nuances, and the specific ways that they communicate these things, and make sure that's included if there is information that would be translated into American Sign Language. Certified deaf interpreters would need to be included in the process to make sure that cultural mediation is there and the message would be understood. There's a lot of health information that deaf participants suggested would be important, as you can see here. One person mentioned that when the healthcare system develops materials that fit the deaf population, that is actually a universal design that would benefit many different populations, not just deaf people. In regards to health information dissemination strategies, participants mentioned it was important to collaborate with the education system and with the community. In regards to community engagement, it's important to not only connect with them physically, but virtually. Physically would include going to deaf clubs and deaf events. Virtually means engaging in social media, being on those Facebook pages and developing relationships with them through those medias. All in all, with the underlying roots and findings of the interviews, we see that deaf people are not included in public health efforts, and they're not considered. There's complicated barriers and frustrations that deaf people go through in order to get accommodations to understand this health information. There's a stigma on deaf people, therefore they are typically forgotten. There are nuances in culture and language that typically are not considered from those who are not part of the community. There are not resources available that are given to the education system to teach deaf children at a young age. In regards to the social cognitive theory, we know that the environment strongly affects this population. So when that is the beginning of the process, they therefore are not able to understand language, and then they're of this health information, and then they're not able to make healthy decisions and beliefs, and therefore it will just have a sequelae on their health literacy later in life. At the end of the day, we see autism is an underlying root issue. Thinking that deaf people cannot go through life because they can't hear, they have a more poor quality of life. This stigma causes this systemic oppression, and coming up with health programs and material, really, we wouldn't need to come up with all of that if we can get to that systemic autism level first and start in the beginning. With the hypothesis, seeing that information is not accessible, if we could come up with materials and that fit the deaf population, there would be an improvement in preventative strategies. We see communication barriers as the root issue from our interviews. Future directions for this would be partnering with the deaf community and public health sectors to have these efforts be engaged. Understanding guidelines on how to appropriately translate information into American Sign Language. With all of these three projects, you can see that it would leverage public health efforts to try to prevent tick-borne diseases. And MEM, the medical ecological model, helps us look at all of those different factors in this study that we've seen, and surveillance programs and how they can improve overall. In sum, with this project, the goal is to improve surveillance programs. And we found Rickettsia parkeri in Ecuador, in a country that is responsible for those diseases in their country that had not been found before. We found 2 different Rickettsia types and 5 different tick species carrying those. We saw a positive interaction with proteins in 3 different kinds of diseases. AIR and GCFP have the ability to detect tick-borne diseases. And with preventative strategies for the deaf population, we see that there is a correlation with the deaf population and public health sectors and how there is no relationship or engagement with them. So we need to have that begin in order to understand their risks, their communication, and therefore their health behaviors. Public health sectors needs to be more inclusive of the deaf population and include captions, have more visual information, hotlines, and resources for the deaf community. The United States is a country that is a monolingual country and has this internalized dominance of being able to hear in one language. So with autism and that stigma, we need to acknowledge that in order to be able to improve the overall system level and access for deaf communities and populations. In closing, all 3 findings in the study saw the importance of using the medical ecological model and the translation process, because there are so many different layers in order to really solve this issue. Thank you everyone for coming. Here's some beautiful pictures from my journey and my studies. It's been a lot of hard work paid off. So I would welcome any questions at this time. So if there's any questions, you are more than welcome to type them in the chat box. I can answer them as they come through. So one interpreter is going to sit next to me and she can copy sign the questions for the deaf participants on the call. So I see a question. I'll go ahead and sign it. It says, how do you think the deaf community in Ecuador is similar or different from the community that you interviewed here in the United States? That's a great question. The deaf population in different countries use their own sign languages. The way language develops depends on that community or country's way of life, attitudes, signs, behaviors. All of that are factors into how their sign language is developed, and that has an influence on how they learn things and how they acquire information. So how people in other countries receive and perceive information is different than we do here in America. So if we did want to apply our findings to those in Ecuador, we would need to consider that. Both deaf populations in Ecuador and the United States use sign language. So that information is important to be able to apply, because information needs to be available in their primary language. Thank you for the question. So we have a question. Carolyn wants to use her video to ask a question. There's one other question first from PJ that I'm going to answer. So she asked, if you had to pick 3 experiences that best prepared you for a PhD program, what would they be? I think it's important to have a lab rotation experience. That really helps you develop your research question to figure out what you want to do for your entire PhD program. The importance of having that skill to identify the right kind of advisor and mentor will help guide you through that long journey that gets you to that point. So a good mentor will make or break you. Third, Third, I think really it just depends on your lab skills and the experience that you bring to the table and that you offer that you can continue to grow on. And the ability to stand up, get up after you fall down, because you're going to fall down a lot during your PhD program. So Caroline? So I'm going to spotlight Caroline so she can ask her question. Hello. Hi. Great. Great to see you, Lauren. Good job today.
Thank you.
Of course, I have an ecology question for you. So in regards to Ecuador, you mentioned land use changes from gold mining historically, the banana export business has affected that. So how have the land use changes impacted the environment for ticks specifically? I'm just thinking if there's more forestation, there would be less animal hosts. So can you explain exactly the correlation on the land use change and the impact on ticks? So I'll turn my video off now. Great question. So Caroline is something I want to add to everyone. She was my undergrad advisor at Gallaudet, and she focuses on environmental science. With land changes, there are 2 that really impact ticks. One is deforestation and agriculture being set up. So there are a lot of factors included, but one of them is the hosts will change their shelters, so they end up moving. And there's a dilution effect. Therefore, the ticks that typically will focus on a host, because there are plentiful of those hosts in that area, will have less chances of disease and transmitting disease. So if there's a specific animal that is infected, and then they all become infected, then they will actually switch their host.
Because there's a large number of animals, there's actually a dilution effect of the pathogen in that population. So there's a lower chance of ticks actually being infected.
So if there's deforestation, they become more specific for the host they're looking for. For example, if there's cows or horses or dogs in that specific area, they could easily be targeted for ticks.
And because there's fewer of them and each animal has more ticks on them, if an animal if a host animal is infected, then there's a much higher chance that those ticks that are feeding from it will also be infected. Also, conservation biologists often want to establish reforestation, which causes problems in itself, because as hosts migrate, a tick-specific host may migrate, but then as things are reforested, the ticks hosts will become more widely available, and ticks will spread from there, and thus spread disease. Ticks are complicated. Again, there are many factors, and environment is just one of these many considerations.
I think we have another question from Alicia. How long have you looked into the antibody responses last to the tick antigens? Would you guess they are long-lived or short-lived?
Good question. It depends on what kind of disease we're looking at. Most typically, IgM and IgG antibodies are used to detect disease response. IgM are more short-lived antibodies. So that's important to know for these different diagnostic tests to include IgG, because those are longer lasting antibodies. So if someone tests positive for that disease or not, the IgG antibodies will last longer and be detected longer. But there's a range of antibodies for different diseases, and some, and like I said, ticks spread over 13 different diseases, which equals 13, that means 13 different antibodies. I have another question.
This is from Zahira. So you did your third project, if you did your third project here in the US, I'm curious how it would be different if you did it in Ecuador. If I did the qualitative interviews in Ecuador, the results would definitely be different, and it really focuses on community-based research, so I would need to connect with that community, learn about them, and work with them. Here in Rochester, with that systemic oppression, we notice that here, and I have that connection with the community here, but United States is known as one of the best countries for opportunities in regards to the deaf population. We have the ADA, which is a law that protects deaf people. Ecuador doesn't have those laws protecting their deaf population, so we would probably see more disparities and no access to information. So we have another question. Climate change will intensify in the future, and disease prevalence may increase in intensity too, especially with COVID-19 and remote communications, having to minimize in-person contact. How do you think the deaf populations around the world will be impacted by our changing climate and our changing needs of human contact in developing and developed countries? And how do we keep them informed and minimize the health info disparity? That's a great question as well. The problem that I mentioned is the digital divide. Many deaf people experience the consequences of the digital divide because of low SES status. They don't have funding opportunities. They have low employment opportunities, so therefore they may not be able to afford technology and get access to that information that is provided on the web. So how can we continue to notify the deaf community of information that's necessary? In regards to the United States, there was one person that said it well. Almost all deaf people have a relay service phone number, the video relay service, a video phone. Therefore, we can use that to connect with deaf populations. One example is Sorensen or Convo. Those are companies that provide video phones. Through that mechanism, we would be able to reach out to all deaf people. those who have low SES and maybe are not economically wealthy would still have a video phone so we could connect with them. It is definitely a challenge, but we're all in this together, so we're going to, you know, that's something that would be a future direction of the study as well. So it looks like Craig said we're getting a little bit long, so we're going to need to hold off on questions and get to the closed door portion. Okay, so someone else said that we can have Jose as the last question. Let's see. Do you believe the time, like a rainy season, that you collected the tick samples could have played a role in your results? Also, how would you recommend to engage with ministries of health globally to attend the social and health oppression that deaf communities are facing? That is a great question. That is why we decided to go to Ecuador in March. That is their rainy season. That is a high humidity percentage in the area, and ticks do depend on the humidity percentage when they start their biological process to look for a host. And therefore, there are more ticks during that time comparing to the dry season. If I were to engage with the ministries of health on a global level, it would be important to engage with the communities, the deaf communities, identify those stakeholders in the deaf communities in Ecuador, and then the Ministry of Health can work with them to have that conversation, and then they can talk later on. So thank you everyone for coming and watching. I hope you enjoyed.
One moment, please. If everybody wouldn't mind just turning on your video. If you're comfortable turning on the video, we could give a proper appreciation to Lorne for his presentation today and congratulate him on his work. So thank you, Lorne. For the committee will be in the other chat room. So thank you, everybody. And Lorne, we'll see you in the other chat room, please.